over, okay?
Perfect. Thanks, Amber.
Excellent.
I really appreciate the invitation to be here today and tell you a little bit about some of the work we've been doing on water intake as a trait for selection in beef cattle. And we're going to slowly kind of work towards whether water intake is a heritable trait towards the end of the presentation. At the beginning here, what I'd really like to do is sort of establish what I think is sort of the groundwork for why we actually should care about water intake as potentially a trait for selection, but certainly in terms of performance of the animal. And then I'll just kind of review a little bit of the literature that's out there, because one of the surprising things when I started sort of thinking about this idea is I thought there would be quite extensive literature already existing on the issue, and there's maybe not as much as you might think, and certainly wasn't as much as I think. So from there, I'll talk a little bit about some of the research that we've been doing in that area. So to get us started, I'd like to just talk really briefly about why we would actually care about water intake as a trait. And I think the most obvious reason, or the one that is probably at the forefront of most people's minds, is that water is one of the 6 essential nutrients. But I sort of think of it as the neglected one of those 6 essential nutrients, because if you think about it, when we balance for rations and think about what we're feeding to animals, a lot of times we're really thinking a lot about what is the protein content of the ration, or are we providing carbohydrates and fat as an energy source. We may even be thinking about delivery of vitamins and minerals to those animals from either a performance standpoint or a health standpoint. But I think water is one we don't often actively have to think about, because it's often just there and we just sort of assume it's there and of high enough quality that we don't have to give it a lot of thought. And certainly that wouldn't be true for all producers, because we have producers in areas of the country that water quality isn't that great and not as available. But I think it's sort of easy to neglect and easy to forget that water is one of those really important essential nutrients that we need to make sure that we have available for these animals. And water intake has, of course, been tied to performance because it is one of those 6 essential nutrients. There hasn't been as much work in this area as I would have thought, but it has been tied to weight gain in the literature, and in particular, the ability of animals to deal with heat stress and other types of environmental challenges. The specific mechanisms that animals use to sort of overcome those challenges related to water are less well defined. Water intake really hasn't been explored very much from a genetic standpoint outside of certain specific water conditions for cattle, like high sulfur as an example, or outside of model organisms like mice and organisms like that. And finally, I think the third reason that we should really give some thought into water intake is that water is not necessarily an unlimited resource, even though sometimes we sort of maybe be tempted to think about it that way. Of course, in certain parts of the country, water is not really limiting within the environment, and may not be as big of a concern in those areas. But we do have a lot of cattle in pretty large portions of the country where we do have less rainfall. And of course, we also have, in many of those areas, we also have issues surrounding use of that water or water rights. Regardless of what portion of the country you're in, you may be affected by drought, and sometimes water intake tends to come to the forefront a little bit more when we're experiencing those types of issues. And regardless of whether we're in an area where there maybe are water challenges or not, we do still sometimes have to deal with the impact of competition. So certainly competition from humans and municipalities that need to use water resources that are available, competition from wildlife in the area, and also competition from other agricultural enterprises like irrigation water used in crop production as an example. And so these figures here show a little bit related to areas of the US that show evidence for water stress, or areas of the country that are maybe predicted to be impacted by changing climate and how that might impact water resources or cause a decline in water resources in those areas. And that brings me to the fact that the importance of water intake when we're thinking about it is definitely going to be variable. And it's going to be variable in sort of two different characteristics, temporally and spatially. So spatially just meaning depending on which part of the country that you're in. If you have lots of precipitation and you have tons of available groundwater that's of high quality, it's probably not going to be as big of an issue for producers in those areas. But we do also have to deal with some of those temporal issues related to drought. So it may not be as critical and sometimes as it is in others due to drought situations. So I want to transition now to kind of thinking about what we know about water intake from the scientific literature. And I'll kind of relate this back to genetics where possible, but I want to kind of give a little bit of a foundation beyond that as well. From a genetic standpoint, we do know that there are breed differences in water intake, and that was established back in the 50s by some work by Winchester and Morris. And that paper is really the basis for the NRC guidelines for water intake. And those guidelines, at least as far as I can tell, are largely based on this work. What you can see from this graph on the screen here is that we have two different sort of cattle populations here, Bos taurus cattle and Bos indicus being tropically adapted cattle like Brahman. And in this case, the European cattle, Bos taurus, I believe, were largely Angus and Hereford, so British-based breeds more than anything. But those types of breeds would make up the majority of the germplasm in the United States. And what we see is that at similar levels of temperature and humidity, Bos taurus cattle will drink more than Bos indicus cattle. We can also see in the literature that cattle will sometimes voluntarily restrict their water intake if water quality is really poor. Sometimes there's even a little bit of restriction even if there's chemically treated water versus not. They'll restrict their intake quite a lot more if we have conditions such as high salt. And we know that if these sort of conditions arise, perhaps within sort of the classic summer drought, that this water restriction impacts body temperature regulation, which could have an impact on heat stress if we've got that classic hot, dry summer drought condition. And we know water intake can be impacted somewhat by water temperature, but that's work in that area is a little bit conflicting. Excuse me. In terms of the amount of water cattle are actually drinking, a lot of the work that's out there is going to be based on large pens of animals in feedlot type conditions. So you put a water meter on the line coming into the pen and you measure how much that entire pen drinks over an entire day and sort of extrapolate that back out to the individual animals. So they're very much sort of average intake values spread across a large number of animals. And we don't have a lot of data that really looks at individual animal variation because it's that pen data. But average intakes in the literature for pen-based data will be around 30 to 40 liters per head per day in the summer. In the winter, there's less data out there in the literature related to winter water intakes, but they'll be around about half that at 17.3 liters per day in this particular study. There's also work out there that looks at predicting water intake, and there's a couple studies that do that based on the environmental conditions that are present in the environment at that point in time. So if we think about individual animal water intake, there's a lot less data available out there, but we do have the technology down to collect large numbers of water intake records on individual animals. And that's primarily through the use of two different systems, either the GrowSafe system or the InSynTech system. Two studies that are out there that have been published in the early 2000s looked at intakes for either lactating dairy cows or Holstein bulls, so both dairy populations, in an InSynTech system. And you can see the ranges on the screen there. They're quite large in terms of the amount of range in that data. The only study that I'm aware of in beef cattle that would look at that on a large number of animals would be this GREW and other study published in 2011, as well as the work that I'm going to show you today. So we'll delve into that in a minute. But this GREW study had a mean water intake of about 30 liters per head per day, so similar to what's been published for summer intakes. These cattle were at kind of thermoneutral conditions. Interestingly, they were able to replicate the finding from the paper in the 50s that showed that those tropically adapted breeds will drink less than British and continental-based breeds. They did not note any difference between bulls, steers, and heifers, but probably because they were fairly young growing cattle at that point in time. If we think about water intake and using individual water intake data from a genetics or selection perspective, there's really not a lot out there in the literature. There are no heritability estimates or genomic studies of water intake in beef cattle that I've been able to find at this point in time. There is some work that shows that it's a heritable trait in mice, and there have been several successful selection experiments to select mice basically for divergent phenotypes. So mice that drink a whole lot of water or not very much water. So we really wanted to know if this was a heritable trait in beef cattle and if this is something that we might potentially be able to target or want to target through genetic selection. So that's basically what we set out to do with what I'm going to share with you today. So in terms of the data that you're going to look at today, will be data on 579 crossbred steers, and they were fed in 5 different groups here. We will have about 800 animals that we've fed here, but in terms of the data that's been analyzed that I have to share with you today, it'll be those first 5 groups. We'll be adding those next 2 groups here in the near future. So we did have complete feed intake data and water intake data collected using an Ensign-Tech system in these animals. They were fed a growing diet, and there were a few management differences in terms of the feed intake data, but all the groups had access to ad libitum water during the time period that I'm going to talk about, which is this baseline time period of the study. So basically, when I say the baseline period, I essentially just mean that it was a 70-day feed intake test using Beef Improvement Federation guidelines at the time the study was conceived. So that 70 days will include all of the feed and water intake. We also had body weights taken every 2 weeks to calculate average daily gain during that time period. There's a lot of other data that we've collected. We won't really have time to talk about most of that today. But just to kind of orient you to the data, I wanted to just give you some pretty simple statistics here. So on the top in the boxes is the information related to water intake. If you want to look at the dry matter intake or the start weights, it's in the table below there as well. And you can see there were some differences in the size of the animals. So keep that in mind when you're kind of looking at the average values. But in terms of water intake, it would have ranged from about 28 kilograms per head per day up to just under 50 per head per day. In terms of the range from about 15 kilograms on the low end, which is a liter if you want a unit of volume, up to about 100 on the high end. So quite a bit of variation in our data as well. Not quite as much as those dairy studies from before in the literature, but quite a bit of variation nonetheless. These do encompass groups that were fed over both the summer and the winter. So you're seeing data from both of those divergent sort of environmental periods in here. So I'm going to take you through a little bit of sort of the process that we've gone through within this study to sort of set up testing procedures, some of the things that we've looked at in terms of the environmental effects on water intake, and then answer our basic question that we started the webinar with, and that is water intake a heritable trait? So the first thing that we needed to do was set up sort of testing guidelines because there are currently no guidelines for the collection of water intake phenotypes. And it's really important as we think about studies for genetics, it requires a very, very large sample size. So even when we have 800 records that we're analyzing, it'll still be a fairly small sample size for doing things like estimating heritabilities and looking for genetic effects and things like that. So it's really important that we establish, that data collection procedures are sort of valid and we're collecting enough data to estimate those phenotypes really well for each animal, as well as sort of establish some guidelines so that when people do this type of work in the future and collect that data, we can sort of combine that data together with similar procedures to do larger scale analyses. So that was basically what we wanted to do here on the first part. And one of the real questions in our mind was related to the test length, because we know that the sort of classic 70-day feed intake tests are 70 days because of the estimation or the collection of average daily gain phenotypes. That's why they're 70 days long. We can collect accurate feed intake data in sort of 35 to 42 days. So the real question is where water intake would fall along that spectrum, how much data we would need to collect. So in terms of procedure, we looked at 7-day averages. So we looked at 7-day averages both from the beginning of the test, for example, from day 0 to day 7, or from day 0 to day 14, as well as from the end of the study, so the last 7 days or the last 14 days from the test. And we basically looked at correlations of those values to one another to see when we had enough data to basically conclude that our phenotype was accurate. And our threshold for that was similar to what Beef Improvement Federation used for setting up guidelines for feed intake. And that, in our case, was a Pearson and Spearman correlation that exceeded 0.95. So the Pearson correlation is telling us that the values are numerically similar, and the Spearman correlation is telling us that the animals are ranking similarly. And when both of those correlations exceeded 0.95, we said, okay, that's how much data we need. And we found pretty consistently that we needed between 35 and 42 days of good water intake data to collect an accurate phenotype. So the great news about that is that we can collect water intake and dry matter intakes simultaneously without making the test any longer, and we can still decouple from those 70-day gain tests if we need to, which is reflected in the new Beef Improvement Federation feed intake guidelines. So sort of the ability to get 2 phenotypes for the amount of time as one here if we have facilities that can collect that data. So from there, we wanted to really look at the effects of the environment on water intake and develop some prediction equations to see how much of the variation in water intake, individual animal water intake, we could explain just based on knowing the conditions in the environment. And that's been done with pen intake data, but hasn't really been done a lot with individual animal intake data. So we really wanted to do that in this study. And if you're not familiar with these types of systems, I threw this picture in here to kind of show you. This is the actual Insim-Tech system. So basically you have feed bunks and water bunks that are on load cells to measure. And then there's a pneumatic gate here that will either allow or disallow an animal access. So when the animal wants to access the bunk, sticks its head through the little stanchion there, the tag is read by the reader, and then the gate will drop, allowing them access to the bunk. Before the gate drops, a weight is read, and that'll be sort of the starting weight for the bunk. When the animal leaves, the weight is taken again, and by subtraction, we can get the amount of consumption of either feed or water for that particular animal. Okay, so if we take a look at the environmental factors for this particular study that we looked at, we had temperature, which ranged from about 39 Fahrenheit to 82 Fahrenheit on average. So you'll see when you look at the minimums and maximums that there were more extreme values than that, certainly within each group, but that's where the sort of mean temperature would be within those different groups. Relative humidity, where we didn't see a ton of differences between those groups. No major differences in wind speed. And then finally, we had solar radiation values, which, as you might imagine, in the summer were quite high, and in the winter were substantially lower. And those were the parameters that we used in our analysis. So here, you have each of the different groups, 1 through 5, and then we also have a contrast between summer and winter, that SP versus WP. So basically, in the part that says baseline that has a box around it, that basically gives you the water intake in % of body weight per day within each of those different groups, so averaged across each group. So you can see there were some pretty substantial differences between those. So the difference between summer and winter in this sort of baseline analysis, where we didn't account for any environmental parameters, was about 2.3% of body weight per day. Now, as you go from left to right in the chart, we're adding different environmental parameters and then reevaluating the differences between groups. So we added solar radiation, added temperature, added humidity, and finally added wind speed, and then took a look at the differences between those again. And once we looked at those differences with those 4 environmental parameters accounted for, the difference between summer and winter intakes was about 0.2% of body weight per day. So I present this to tell you that I think we had a pretty good handle on the environmental factors, because we were able to sort of reduce the differences, mean differences between those groups by adding these 4 environmental parameters. So I think we had pretty good data and the right environmental parameters in the analysis here. So with that, we developed prediction equations to predict water intake. So on the left, you'll see the overall, followed by the next column will be the summer and then the winter. And I'm not going to worry about anything on the right-hand side of the slide today, just for time purposes. So you can see our main drivers of predicting water intake in the summer are dry matter intake and the average ambient temperature, whereas in the winter, it's pretty much solely driven by dry matter intake. Interestingly, when you combine all the data together, the biggest model drivers are dry matter intake, the weight of the animal, and then the average temperature. And if we take a look at all of that, we see the R squared there on the bottom is 0.4. So when we account for all of those different things, dry matter intake, the weight, and then our 4 environmental parameters, we're able to explain about 40% of the variation in those individual water intakes. So from that, we concluded that you can make predictions if you have weather factors and dry matter intake of the water intake of animals. But as a geneticist, what's really interesting to me is that we have a lot of individual animal variation that we are not accounting for when we are looking at differences in dry matter intake, weight, and weather factors. And that makes me think that maybe we have a heritable trait here that has some genetic variation underlying the trait that we could select for. And if it is heritable, the other thing we really need to understand is what are the relationships between water intake and other economically relevant traits to understand if it's something where we would have negative impacts on other traits if we were to select for it. So that's where I want to spend the remainder of the time that I have today talking about. So really characterizing water intake as a phenotype. So we have the water intakes and dry matter intakes, and we've talked about those. We also calculated a few other variables here for both the feed data and the water data. So we have a feed to gain ratio and a water to gain ratio. And we also did a residual feed intake calculation using average daily gain and metabolic midweight, and a residual water intake calculation using dry matter intake and metabolic midweight. We've sort of referred to these as calculations here as efficiency metrics. I think we could maybe make an argument that they don't really reflect efficiency very well, but for the sake of argument, that's what we're going to call them today. So if we plot those out, here you've got water intake on the y-axis versus a variety of different traits on the x-axis here. So on the top left, you have dry matter intake, and go one panel over, it'll be residual feed intake and then feed to gain ratio. What we see with sort of, I'll call them the feed associated traits here along this top part of the slide, we see that the highest relationship is the relationship between water intake and dry matter intake. And you see kind of a moderate relationship between those two variables. The relationship between water intake and residual feed intake and feed to gain ratio is a little bit lower for both of those as you move from left to right. As you might imagine, the relationship between water intake and water to gain ratio and residual water intake is much higher. Interestingly, if you look at the bottom right-hand panel, one of the things we were really interested in was the relationship of water intake and average daily gain, and I thought there would be probably a substantial relationship there, and what we actually saw was basically no relationship. That line is pretty flat. So we really didn't see a lot of strong phenotypic relationships between water intake and any of our sort of feed-related traits or average daily gain, where we saw basically no relationship. We did see moderate relationships between water intake and the other sort of water-related traits that we calculated. So to sort of take this a step further and sort of get at heritability estimates, one of the things, you need one of two things. You either need a pedigreed population or you need genotypes. In this particular instance, we did not have a really solid, well-defined pedigreed population. So instead, we went the route of genotyping. So they were genotyped using basically the GGPHD 150K chip. If you're not familiar with that product, basically what it does is it gives us genotypes back or data back on 150,000 different single base changes within the DNA. So spread throughout the genome. So we had 150,000 data points there in terms of our genotypes. The other thing that we knew we had in this population was a lot of admixture, a lot of crossbreeding. And because we know that breed composition can impact water intake, we wanted to make sure that if we had some of those other breed types in there, particularly Brahman as an example, any of those tropically adapted breeds, we wanted to be able to account for that in the analysis. So we did estimate breed composition with our SNP data because we didn't have that available through pedigree. So we used a regression-based methodology, and a big thank you to Larry Keene from the U.S. Meat Animal Research Center for making allele frequency data available to us to be able to do that. But what you see is the result of our breed composition analysis here on the screen. So when we went through and enrolled animals in this study, we obviously didn't know the exact breed background on a good number of those, but we did try to actively select against visually, select against those that we thought looked like they might have Brahman ancestry or dairy ancestry. And you can see we were pretty successful in doing that. The largest breed composition in all of the different groups was Black Angus. You can see we had a lot of other Bos-Taurus-based breeds that were included in that in all those different groups. You can see in the first three groups we had a little bit of low level of Brahman influence, which makes sense because those cattle came from the southern United States. So even though we tried to filter that out, we weren't completely successful in those first three groups. And in all the groups, we had a little bit of very, very low level dairy influence. So we included that in our analysis to basically adjust out any potential effects on water intake. Because we didn't feel like we had any breed groups other than Angus that were really large enough to do a good job estimating breed effects, what we decided to do was just basically group them based on biological type. So into British and continental breeds, and then we had groupings for and covariates for Indicus and dairy ancestry there. And basically, again, our goal here was really to, since we couldn't estimate the breed effects, is really to remove the impact of any sort of residual dairy in the Indicus germplasm within the population to the extent that we could. This slide is ugly, I apologize for it, but I promise we won't have to go through the equation. I just wanted to leave it up here to point out that we did do a genomic BLUP using the a relationship matrix derived from the SNP data. So basically, just like generating regular EPDs, only we did it with the genomic data instead of pedigrees. We did fit these as bivariate models, so 2 traits at a time, essentially. We had some effects in there for group and feed management. We also had covariates in there to account for the weight of each animal as well as those different ancestry percentages. And with that, I can finally answer the question at the beginning of the webinar, which is water intake a heritable trait? And the answer is yes, it's a moderately heritable trait. So in the very top line on the far right-hand side of the top chart there, you'll see the heritability for water intake is 0.39. So pretty on par with what you typically see in the literature for growth traits and things like dry matter intake, things like that. So all of our water-related traits, which I've sort of denoted with those arrows in that chart, so the top line, water intake, as well as residual water intake and the water-to-gain ratio, were all kind of moderately heritable, about 0.37 to 0.39, so pretty narrow range for those heritability estimates. Now that we know the heritability estimates, one of the other really vital things to begin exploring is the genetic correlation of water intake with other traits. So the genetic correlations will basically tell us if we select for water intake, what changes we are likely to see in other traits that are genetically correlated. So that's what I've boxed here on the top level, top line of the table on the bottom part of the slide. So as you might imagine, the genetic correlation between water intake and our sort of water-derived traits, residual water intake and water to gain, are quite high. Nothing very surprising there. We also notice that the feed to gain correlation is quite high. But I want to point that one out as a bit of an aberration, because the standard deviation that you see there is sort of outside of the accepted parameter space. So I think we have a little bit of an issue fitting that trait because of the fact that it's a ratio trait, and also because it's a fairly small sample size. So I would take that feed to gain value with a little bit of a grain of salt. We also see that the genetic correlation between water intake and residual feed intake and dry matter intake are just a little bit more than 0.3. So a fairly small but positive relationship between those. So as water intake would go up, we would expect dry matter intake to also increase and residual feed intake to also increase. Interestingly, the genetic correlation between water intake and average daily gain was basically 0, which corresponds with our phenotypic correlations, but not necessarily something that I expected we would find at the beginning of this study. So water intake and water efficiency traits are moderately heritable. We didn't note any strong antagonistic relationships between water intake and any of our feed intake traits, and really no relationship with average daily gain. So the big question now is there are certainly more economically relevant traits than just dry matter intake, as an example. So we really need to sort of expand this analysis to some other traits. And we do have the data to do that in the case of carcass traits, and we've done that analysis, so I thought I'd share that with you today. And just the little bit of time I've got left here. So we did collect carcass data on all of these animals. So when I say carcass data, we have hot carcass weight, ribeye area, back fat, marbling, and yield grade. In this analysis, you'll also see final body weight. So that was final body weight taken within 24 hours of harvest. We did have animals go to a variety of different plants, so we did account for this in our models. We also accounted for differences in days on feed during the finishing period for each of these animals. You can see some spent quite a lot of time in the finishing period, some not very much at all. So we did account for that in these analyses. So if we just look at sort of these raw phenotypic correlations or relationships, what you can see is we've got water intake on the x-axis now and our different carcass traits on the y-axis. And you can see that all of those lines are pretty flat. And so there's really uniformly weak phenotypic relationships between water intake and carcass traits. So the question now is, what do the genetic relationships look like? So we took a look at the genetic correlations. And I want to point out, similar to the values that you looked at on that previous slide, this is a really small data set for doing these estimations. So you see that these numbers in parentheses tend to be quite large. That's because of the small sample size. So we would expect as we get more data into this analysis and as we collect more data into the future, that we'll do a better job estimating these genetic correlations. So you can see most of them, nothing really super high. The two highest were with hot carcass weight and back fat. The standard deviations would suggest that those are not different from 0, but in this case, if we just take the numbers at face value, we can see that fairly low levels of relationship between those. Sort of in the next tier down would be the relationship between water intake and yield grade and final body weight at 0.29. Again, large standard deviations there. The next level down would be marbling at 0.17, and then finally, very small relationship between water intake and ribeye area. So the genetic correlations between water intake and the carcass traits that we were able to measure in this particular data set were basically 0 to slightly positive, although we did have those large standard deviations to keep in mind. Potentially some slight antagonisms with hot carcass weight, marbling, and final body weight, but again, you need to have more data to get really precise estimates on these to understand the impact of those. But just to kind of wrap this up, the test length that we really need to collect water intake data is pretty compatible with decoupled feed intake and gain tests. So potential for collection of 2 phenotypes for the price of 1, if you will, if the facility has the technology to collect water intake. Water intake can be predicted with moderate accuracy for the quote-unquote average animal. But there is a lot of individual animal variation that we really can't model well with environmental and performance characteristics, which leads us to the fact that that individual animal variation is important, and water intake is a heritable trait. When we take a look at its relationships with other economically relevant traits, we see that antagonistic relationships are generally low to moderate and probably wouldn't seriously hinder selection efforts, particularly if we could figure out a good way to put these traits within a selection index. Economic selection indexes are generally the best method for selection on any type of multi-trait selection for economically relevant traits. So ideally, we could include it in something like that. However, just to kind of take us back to the very beginning part of the presentation, within an economic selection index, one of the things we have to do is weight the trait based on its economic importance. And the economic importance of water intake will be probably pretty hard to establish, and it probably isn't going to be economically important in certain regions of the country. So one of the things that may need to be developed to really use this trait well within a selection index would be customizable selection indexes, so that producers could sort of have input on water costs or the importance of water within their operation. So with that, I have a lot of acknowledgements that I need to make. We had collaborators both here at Kansas State and the U.S. Meat Animal Research Center that were instrumental in the data analysis. We also have individuals at a variety of different institutions that were involved in the original grant that funded this work from the USDA. We have graduate students at Oklahoma State University that did a lot of the data collection and really contributed a lot to sort of the phenotypic data collection side of this project. And we have some graduate students at Kansas State that are working on data analysis in this project. And most notably, I want to make sure that I highlight Cashley here. So that's her picture on the screen. Cashley is a PhD student of mine, and she finished her degree last year. But she did all the analysis that you're looking at and that you saw today, and that is in the papers that we've published on this thus far. So she did a great job with this, and wouldn't have much of a presentation without her. So I wanted to make sure that I acknowledged her efforts here as well. Also want to thank, obviously, the USDA for funding the project, and we also received a little bit of supplementary funding from Texas Cattle Feeders Association to do some water quality monitoring within the facility. So I wanted to acknowledge that as well. With that, be happy to take any questions that we have, and thank you all for tuning in.
Excellent. Thank you so much, Megan. So hi, everyone. For those of you who joined late, I'm Amber. I'm the project manager for Grazing Cap. I have a sort of basic question. If you guys have questions, feel free to use that chat function at the bottom of the Zoom window, and I can read those to Dr. Rolfe. So, okay, I'm coming at this. I'm an anthropologist. I'm thinking about the practical application of this, right? So you can measurement in these pretty complicated facilities, but say I'm just a typical rancher person. Is there any practically apply, how do I look at, say, the set of cows I have, and is there anything I can do to try to sort of optimize their water intake? Do I just pick the ones that appear to be drinking the least amount of water, right? Because I'm assuming most ranchers aren't going to have this sort of setup to actually measure individual consumption.
Absolutely. So not a basic question at all. Great question. So there's a couple of different routes to take, I think. So one is that there are different bull tests and things like that that have feed intake testing available. If those facilities incorporated water intake testing as a part of their services, any producer could enroll cattle in those facilities or purchase bulls that have been tested within those facilities to be able to get water intake data on sort of important animals within the herd. And if we're thinking about commercial producers, really you're probably not going to be measuring your cows. That's not going to make any kind of economic sense. But one thing that makes sense for commercial producers to focus on is bull selection. So if we have some data available on a bull, Of course, when we keep replacement females out of that bull back into our herd, we can expect that over time, and on an average, we've done a good job of selection of those bulls. Those characteristics would be reflected in the cows as well over time. So that's one way to go about doing it. The second way is that one of the really awesome things about having national cattle evaluation and EPDs available for selection of typically bulls for commercial cattlemen, but also potentially females as well, is that sort of the seed stock sector collects data on some of these traits and can use that information that comes from producers that are used to collecting a lot of really complex phenotypic information and doing a really good job at that, and to be able to develop selection tools like EPDs on these types of traits. And then nobody's necessarily limited to phenotypic selection on that. They have sort of a better tool to look at genetic selection on those types of traits. So there's a couple different ways to go about this. I think focusing on bull selection, hopefully someday with an EPD if the industry decides that this is something that's important to sort of go after. And that'll make that really feasible even for you know, everyday commercial producers that may not have access to those types of facilities.